All right, let's dive right in. Today, we are taking a deep dive into the incredibly complex world of the basal ganglia and the internal capsule. Now, these are two structures buried deep inside our brain that are, and I mean this, absolutely critical for every single move we make, from the tiniest little gesture to the most complicated action. So let's get started. So, to really get a handle on the basal ganglia, first we've got to see where they fit into the bigger picture. This diagram here lays out the brain's entire motor control system. You can see up at the top you've got the command center. That's the upper motor neurons in your motor cortex, planning and directing all of our movements. Then the cortex checks in with two key advisors, the cerebellum, which handles coordination, and the star of our show, the basal ganglia. And notice the description here. It says they're involved in gating the proper initiation of movement. That word, gating, is perfect. It suggests they're like a gatekeeper, deciding which movements get the green light and which ones don't. So the big question we're asking today is, how exactly do they pull off this critical gatekeeping job? Here's the game plan for our session. We're going to kick things off with a big picture overview of these motor gatekeepers. Then we'll get into the nitty-gritty anatomy of both the basal ganglia and the internal capsule. After that, we'll trace the flow of information through their circuitry, see how they fit into the wider extrapyramidal system, and finally, we'll shift our focus to the really powerful clinical side of things. What happens when this whole system goes wrong? Okay, section 1. Let's start with that core function. If you think of the cortex as deciding what to do, you can think of the basal ganglia as helping decide how and when to do it. They really are the brain's motor gatekeepers, making sure our actions are smooth, purposeful, and well-timed. So what's their actual job description? Well, first and foremost, they fine-tune our voluntary movements. They're also heavily involved in learning new motor skills and selecting just the right motor pattern for whatever we're trying to do. But the real magic is how they do it, and it all comes down to this really sophisticated feedback loop. Here's how it works. Information flows from the cerebral cortex to the basal ganglia. The basal ganglia then process it, tweak it, make adjustments, and send that refined plan over to a relay station called the thalamus. The thalamus then sends this newly edited version right back to the cortex. It's only after getting this adjusted feedback that the cortex gives the final go command to the muscles. This whole loop is what keeps our movements controlled and precise instead of just random and chaotic. All right, to really understand that feedback loop, we have to get to know the key players involved. So let's move on to the anatomy and meet the 5 core nuclei that make up the basal ganglia. Okay, this slide right here, this is basically our cheat sheet for the next few minutes. The basal ganglia are made of 5 pairs of nuclei, but they're often grouped together based on their location or what they do. Now, I want you to pay really close attention to 2 of these groupings. First, the striatum. The striatum is just the caudate nucleus plus the putamen. Simple enough. Second, we have the lenticular nucleus, which is the putamen plus the globus pallidus. And yeah, you're not seeing things. The putamen is part of both groups. It's a member of both clubs, which might seem a little weird now, but it'll make perfect sense once we start looking at the circuitry. So keep this little chart in your head as we break down each piece. So here they are, the 5 core nuclei. We have the caudate nucleus, the putamen, the globus pallidus, the subthalamic nucleus, and the substantia nigra. Now together, these form a group of subcortical nuclei, which is just a fancy way of saying they're located deep inside the brain beneath the cortex. They're a major part of what's called the extrapyramidal system, which handles our more automatic background movements. And with all their connections to the cortex and the thalamus, their ultimate job is something called action selection. Basically, at any given moment, they're helping your brain decide which of the many possible things you could do is the one you're actually going to execute. First up, let's talk about the caudate nucleus, a major part of the striatum. It's this really elegant, long, C-shaped structure that curves right alongside the brain's fluid-filled lateral ventricles. Anatomically, it has a head, a body, and a tail. And what's really fascinating is that its tail goes on to connect with the amygdala, which is the emotion center of the brain. That little connection is a huge clue that our movements are not just purely mechanical, they're deeply tied to our emotional states and motivations. Functionally, the caudate helps regulate our body and limb posture, and it also plays a big role in goal-directed behaviors, learning, and emotion. Clinically speaking, it's been found that an underactive caudate nucleus has links to conditions like depression and schizophrenia. And this slide gives us a fantastic look at its structure. Over on the left, you've got a coronal view. That's like a slice from front to back. And you can see the caudate nucleus sitting just above and to the side of the thalamus. But it's the 3D view on the right that really brings its beautiful C shape to life. You can see exactly how it wraps around the thalamus and the putamen. Seeing this spatial relationship is so important for understanding how all these deep brain structures talk to each other. Next up is the putamen. Now remember, this is the one that's part of both the striatum and the lenticular nucleus. It's a large, round structure, and it holds the title for being the most lateral, or the outermost, of all the basal ganglia. As we just saw in that 3D image, it's basically nestled right inside that C-shaped curve of the caudate nucleus. And just to get our bearings, on its inside, or medially, is the globus pallidus, and on its outside, laterally, is the external capsule. Okay, moving inward from the putamen, we find the other half of the lenticular nucleus, the globus pallidus. Now this isn't just one single structure. It's split into two really important segments. There's an external part, which we call the GPE, and an internal part, the GPI. And trust me, this division into external and internal isn't just some random anatomical detail. It is absolutely fundamental to how the basal ganglia circuits actually work, and we'll come back to that in a bit. Its main job: to regulate our movements so that they're smooth and precise. Here we can see exactly how the putamen and globus pallidus fit together like a puzzle. The image on the left shows the larger putamen on the outside, with the globus pallidus just nestled up against its inner surface. Then, the coronal view on the right highlights this entire combined structure in green. When you see them together like this, they're known as the lenticular or lentiform nucleus, which makes sense because they have this kind of lens-like shape. All right, let's talk about a structure that's not technically in the same neighborhood, but is functionally a core member of the basal ganglia team, the substantia nigra. Its name literally means black substance, and it gets that name from the huge amount of neuromelanin in its neurons, a pigment that's actually related to the melanin in our skin. It's divided into two incredibly important parts. First, you have the pars compacta, which contains the neurons that produce dopamine. It is the death of these specific cells that leads directly to Parkinson's disease. The other part is the pars reticulata, and its job is to help control eye movements. This slide gives us a great close-up view. On the left, in that detailed cross-section of the midbrain, you can clearly see that dark band that is the substantia nigra. The labels are pointing out its two sections: the dorsal or upper, pars compacta, and the ventral or lower, pars reticulata. The schematic on the right just gives us a cleaner, simplified diagram to help lock in where these pieces are in relation to everything else in the midbrain. Okay, so we've met all the core nuclei, but that leaves a big question. How do they actually talk to the cortex? Well, for that, we need to understand the information superhighway that they all rely on, which is called the internal capsule. So the internal capsule isn't a nucleus. It's a massive V-shaped bundle of white matter, which just means it's packed with nerve fibers. You can think of it as the main freeway for almost all motor and sensory information that's traveling to and from the cerebral cortex. It's organized into three distinct parts. First, the anterior limb. This part contains frontopontine fibers, which go from the frontal cortex to the pons, and thalamocortical fibers, from the thalamus to the frontal lobe. Then you have the genu, which literally means knee. It's the bend in the V. This part carries the cortical bulbar fibers from the cortex down to the brainstem. And finally, there's the posterior limb, which is jam-packed with the super important corticospinal fibers controlling voluntary movements, as well as all the sensory fibers that are traveling up to the cortex. Right, so now we know the individual structures and we know about the highway that connects them. We're finally ready to map out the flow of information. This next section is all about the circuitry, the specific paths that signals take as they enter the basal ganglia, get processed, and then exit the system. So where does all this information come from in the first place? Well, this diagram shows us the main input routes. You have signals coming from pretty much the entire cerebral cortex, the frontal, parietal, and temporal lobes, and they all converge on the striatum, which you'll remember is the caudate and the putamen. These pathways, as a group, are called the corticostriatal pathways, and they are the primary on-ramp for information getting into the basal ganglia system. Let's trace the entire circuit step by step. As we just saw, the main inputs comes from almost the entire cortex into the striatum. The primary visual and auditory areas are the main exceptions here. Okay, so from the striatum, the signals then get sent to one of two main output nuclei, either the globus pallidus or the pars reticulata of the substantia nigra. Now, this fork in the road is super important because it leads to two very different output pathways. The first one, through the globus pallidus, sends signals to the thalamus. The thalamus then relays that information back to the motor cortex, and boom, that completes that crucial feedback loop for fine-tuning our movements. The second output path, going through the substantia nigra pars reticulata, connects to neurons in a structure called the superior colliculus, and its job is to specifically regulate our eye movements. Okay, now that we've walked through the internal wiring, let's zoom out a little bit and place all of this within a broader system we mentioned right at the beginning, the extrapyramidal system. This is going to help us understand the kind of control the basal ganglia really have. The extrapyramidal system is basically in charge of all the involuntary and automatic control of our bodies. We're talking about things like muscle tone, balance, posture, all the stuff we do without consciously thinking about it. It's the reason we can stand up or walk down the street without constantly focusing on not falling over. And as you can see from this diagram, it's a whole network of structures. The basal ganglia are central hub, but they work together with the cerebellum, the red nucleus, the substantia nigra, and the reticular formation to get the job done. And that brings us to our final and maybe most important section, the clinical side of things. Now that we've gone through all the intricate anatomy and circuitry, we can finally appreciate what happens when this incredibly complex control system breaks down. Because when there's damage to the basal ganglia or their connections, it results in a whole range of really debilitating movement disorders. When the basal ganglia aren't working right, the symptoms usually fall into two main categories. Over on the left, we have what we call positive signs. Now, these are unwanted, involuntary movements that suddenly appear. This includes things like a tremor, especially when at rest, athetosis, which are slow, writhing movements, chorea, which are fast, jerky movements, bowlism, which are violent, flailing movements of the limbs, and dystonia, which are sustained, painful muscle contractions. Then on the right, we have the negative signs, which are basically an impairment or a loss of normal movement. This includes akinesia, a total lack of movement, bradykinesia, which means slowness of movement, a classic shuffling walk, difficulties with speech, and a loss of postural reflexes. This slide shows a really stark visual contrast between the two most famous basal ganglia disorders. On the left, you're looking at a cross-section of the midbrain from someone with Parkinson's disease. If you look closely, you can actually see that the dark pigment in the substantia nigra is faded. That's where the dopamine cells have died off. Then on the right, you see a brain slice from a patient with Huntington's disease next to a healthy brain. The difference is just devastating. Notice the severe atrophy or shrinking of the striatum. That shrinking, in turn, causes the fluid-filled lateral ventricles to balloon out and become massively enlarged. So the key takeaway here is that these very different diseases happen because of damage to different parts of the basal ganglia. Parkinson's disease is caused by the death of those dopamine-making neurons in the substantia nigra. So it makes sense that the treatments focus on either replacing that dopamine with drugs like L-DOPA or using deep brain stimulation to electrically correct the faulty circuit. Huntington's disease, on the other hand, is a genetic disorder that causes the progressive destruction of the striatum, the caudate, and the putamen. For that, treatment is currently about managing symptoms, using drugs like xenazine to try and suppress those uncontrollable chorea movements. It's also really important, especially in a clinical setting, to know the difference between extrapyramidal diseases and extrapyramidal syndromes, which are often just called EPS. Diseases like Parkinson's, Huntington's, or Tourette syndrome are primary problems with the central nervous system itself. Syndromes, however, are collections of symptoms that are usually side effects of medication, most often from the long-term use of certain antipsychotic drugs. These syndromes can look a lot like the diseases, but their cause is external. And this slide gives you a look at the four major types of extrapyramidal syndromes. First, we have pseudo-Parkinsonism, which, like the name suggests, mimics Parkinson's, with a stooped posture and a shuffle. Then there's acute dystonia, which involves severe muscle spasms and facial grimacing. Next is akathisia, which is this really profound and awful feeling of inner restlessness. And finally, there's tardive dyskinesia, which is marked by repetitive, involuntary movements, especially around the mouth, like lip smacking, chewing, or the tongue protruding. So we'll end on this thought. We've journeyed through the incredibly detailed anatomy and the complex circuitry of the basal ganglia, this remarkable system that fine-tunes our every single action. The question all of this leads back to is, what does it truly mean for a person when this delicate control system goes wrong? As we've seen, damage that is microscopic, deep inside these brain structures, can show up as profound, life-altering changes in how a person not only moves, but also how they feel. It's just a powerful reminder of the direct, unbreakable link between this basic neuroscience and the human experience itself.